So we're going to just try to finish up chapter 15 here. So what have we already talked about? We've talked about how proteins get made in the cytosol and enter into mitochondria, chloroplast, nucleus, or how they get made into the endoplasmic reticulum and trafficked to the cis side of the Golgi, processed through the Golgi, and come out of the trans side of the Golgi. And the default pathway would be exocytosis from the cell or into the cell membrane. So that's what we've talked about so far. So just remember, you can watch this video on how mitochondria works. Watch a video on how things are trafficked through the Golgi, enter into the cis side, processed through the Golgi, and exit through the trans side. So you can watch these videos, and they will obviously help quite a bit with your understanding of how things are trafficked. They can also be trafficked mitochondria or through the endomembrane system or into the nucleus. So don't forget about RAN GTP, RAN GDP, and the movement into the nucleus. So the last things that I wanted to talk about was exocytosis. There's regulated exocytosis and constitutive exocytosis. And then I want to talk a little bit about endocytosis and pinocytosis and how that is going to help us with recovering some of the membrane as well as taking in things from the outside. So exocytosis is part of the default pathway of secreting proteins out of the cell. Many proteins, their job is to secrete stuff. An example of that is when we were talking in chapter 13 and 14 about insulin. Insulin is a gene product, and so it gets made in the endoplasmic reticulum, and it goes through a regulated exocytosis from the beta islet cells into the bloodstream. And so it's important that we understand how exocytosis works. So, like I said, there's two forms. There's constitutive and there's regulated. Constitutive exocytosis is the default pathway. The proteins are made in the ER, processed through the Golgi, and then trafficked via vesicles to the cell membrane. And then there's regulated. And we've talked a couple of times about regulated. We talked about like I said, the insulin, and we also talked about in nerve cells, those synaptic vesicles don't release their neurotransmitters into the synapse unless there is a membrane depolarization that allows for that calcium to come in and bind to the synaptotagment and cause the V snares and T snares to corkscrew up. So there's basically two pathways. The unregulated, this would be for things like extracellular matrix proteins or proteins that are regularly made and secreted. And then there's a regulated secretion, like in neurons, a neurotransmitter binds to a receptor, causes a membrane depolarization, and a subsequent release of synaptic vesicle contents. So these secretory vesicles will dock and get primed and sit there waiting for the signal from outside of the cell to release their contents. The way that vesicles bind to the membrane. We talked about this before, but I just wanted to remind you, every vesicle has V snares on it. That's where the V comes from, vesicle. And snare is an acronym of an acronym. So it has to do with a protein called NSF that breaks apart the snare proteins after they've twisted together. And so this is an NSF associated receptor. So the snare is an acronym, and the V snares are on the vesicles, and the T snares are on the target membrane, and they're kind of some alpha helices that are sticking out into the cytosol, and so through a coil kind of interaction with each other, they just get tighter and tighter, like two corkscrews winding together, or two threaded fasteners, like a nut and a bolt. And so what that's going to do is that's going to pull the vesicle membrane closer and closer to the target membrane. And once all of the water is excluded from between the two membranes, then the two membranes, they just fuse together. And so there are ways to transport into the cell. There's endocytosis and phagocytosis and pinocytosis. So endocytosis usually has something to do with receptors, so that's why we call it receptor-mediated endocytosis. And so there's some kind of cargo that you want to take up. So in this case, it would be like the LDL receptor is binding to the yolk protein. So once it binds to its target, this receptor is then going to recruit clathrin. And clathrin is a triskelion-shaped protein, and so when a bunch of clathrins come together, they're going to make something similar to a geodesic dome. They're going to create more and more curvature. So you can just think of this as like this triskelion protein. When they come together, they form something similar to a hexagon. And if you sew a bunch of hexagons together, what do you get? A soccer ball. And so they're forming this soccer ball shape more and more and more until they have a complete clathrin. So it starts out as what we call a clathrin-coated pit, and as more and more clathrins are recruited, it becomes kind of ball-shaped, and so an entire vesicle. The last step to pinch off that last little piece is another protein called dynamin. Dynamin is the one that I talked about in class that the Shibiri flies didn't have, and so those Shibiri flies were not able to do the last step of their receptor-mediated endocytosis, and so they lost all of the vesicle-associated proteins to the cell membrane. And so dynamin pinches off that last little bit and releases the vesicle to the inside of the cell. So once that vesicle is floating around in the cytosol, the clathrin releases, and so we call that process uncoating. And so once the vesicle no longer has a clathrin coating on it, it can now fuse with other membranes using V snares and T snares. And so very often when you bring something in using receptor-mediated endocytosis, you have that clathrin-coated pit becomes a clathrin-coated vesicle after it decoats. it will be a vesicle with some sort of adapter protein on it, some sort of receptor protein on it that targets it for the endosome, which is a larger. It's just a collection of endocytic vesicles, and so these vesicles come together, fuse together until they build something larger called an endosome. And we don't want to digest all of the receptors, and so we'll bud those off, and they can go back through the constitutive release pathway back to the cell surface, and now all we have is an endosome full of whatever the cargo was. And so that cargo and vesicle will, or endosome, will then bind to a lysosome, and the lysosomal enzymes will digest whatever the cargo was that you endocytosed. The lysosome is full of a whole bunch of enzymes. These enzymes could digest all of the proteins of the cell, and so as a kind of a defense mechanism against maybe a lysosome would pop and all of those enzymes would go and digest everything. All of those enzymes only work at an acidic pH, and so we use proton pumps very similar to the ATP synthase, but running in reverse. And this proton pump is going to acidify the inside of the lysosome to make those enzymes work. If the lysosome ever popped and its contents spilled out into the cytosol, those enzymes would be rendered ineffective because it's at the wrong pH. Okay? So inside of the lysosome we have every single kind of enzyme for digesting stuff. We've got things to digest fats, proteins, and sugars, and DNA, RNA, and so we can kind of think of this as like a little stomach for the cell, digesting all of the stuff that you take in, and getting all the pieces parts out of it so that you can then use those pieces for other anabolic processes. So the lysosome has to have all of the enzymes in it. We talked a little bit about lysosomal storage disease in the last video. If it doesn't have the correct enzymes, we could have something serious like one of the lysosomal storage diseases is Tay-Sachs, where you don't have the enzyme for digesting a very specific lipid, and so those lipids just accumulate in the lysosome, and they can become cytotoxic just because of the cell gets all clogged up with these full lysosomes. So these lysosomal storage diseases can lead to neurodegeneration, and Tay-Sachs disease is a very sad disease. These kids die at a very young age. So let's go ahead and move on to the next video.